All right. I apologize for the great delay. Technology is wonderful except when it isn't. All right. This chapter is short and it looks misleadingly simple. It has some things that refer to what you've already seen both in this course and in other contexts. But basically everything in this chapter is something you should know by the end of the chapter. It's all going to be groundwork for about 10 more chapters. Well, 8 more chapters. So it's really worth paying attention to and getting it completely under your belts because everything in it will have multiple applications later on. How do I get rid of this thing? Okay. There we go. Can we get rid of that? What is that thing? Exit. There we go. Metabolism. So metabolism is the turning over of things. It's the making and breaking of things. And there's 2 broad domains of metabolism. Catabolism, in which you take big stuff and make it small and get energy out of it. And anabolism, where you take small stuff and put in energy and make big stuff. And they're easy to keep straight because you've all heard of anabolic steroids, and anabolic steroids get their name because they favor the body's processes towards anabolic things, building up stuff. There are, broadly speaking, several ways to run metabolism, or what we call trophic strategies, what you eat to make a living. The very first ones to do this were cheme autotrophs, organisms that could get their own energy, autotrophs, from chemical sources, like rocks and deep sea vents and sort of nasty tasting things like that. About 4 billion years or so, 3.5 billion years or so ago, a brilliant cell came up with the idea of capturing light energy, and since then we've had photoautotrophs in the world who can capture light energy and turn it into chemical energy and drive their own metabolism that way. But a lot of the stuff that's big and moves around is actually what we call heterotrophs. They eat other things. We eat plants or animals. Wolves are heterotrophs. Slime molds are heterotrophs. Anything that eats something else to get its energy is a heterotroph. So what are the sources of energy available? And this is true both for the photoautotrophs and the heterotrophs to different extents. Pretty generally, we're going to see that the energy is available in polymeric precursors. So for example, if we look at starch, this is just 2 subunits of a huge starch molecule or a huge glycogen molecule. And a very common strategy for breaking down polymeric precursors is to hydrolyze them or break them with water. That's what hydrolyze means, break with water. And so this bond is going to have a water molecule added across it. And now you can see that that bond has been broken. What was here as an oxygen in the bond is now an alcohol group. And what was here as in a bond to an oxygen is also an alcohol group. So one of them picked up the hydrogen, one of them picked up an oxygen. A hydrolytic reaction also can break down proteins. So here we have a polypeptide backbone. Here's the peptide bond. Adding water across it gives us the carboxylic acid and the amine group. Here's a triacyl glyceride, glycerol backbone, 3 fatty acyl tails. This is the bond that can be hydrolyzed, and it would release for a free fatty acid in the process. So when we heterotrophs eat things, that has to happen to make the big stuff small enough for us to be able to take them up. And then what happens during the absorption process? Well, our guts are engineered or differentiated to absorb things in part through, well, that's interesting, in part through active transporters. And we talked a little bit about the glucose indirect and active transport in the gut. There are also active transporters that spend ATP to take up oligopeptides, short pieces of proteins that haven't been taken clear down to amino acids. There are passive transporters that we've seen. A few things can come in simply by passive diffusion through the membrane with no transporter. All of this stuff gets gathered together and passed up to the liver, and the liver then will sort of sift through all that stuff and process it appropriately for whatever the current status is for the person or the organism. This is cholesterol stearate. You can see here the cholesterol has a fatty acyl tail to it. That would need to be, that as a whole is one of the things that can be used as a detergent to help solubilize things. Now, we're not eating all the time. So there must be some way to store energy, and there are several ways to store energy. The two principal ways of storing energy are triacylglycerols, that three fatty acyl tail off of glycerol, or glycogen. And glycogen is sometimes called animal starch. This is a very schematic picture of glycogen. It's more heavily branched than the starch that comes from plants, but it's chemically similar in terms of the nature of the bonds that connect the glucose subunits. And we'll see that in more detail in the carbohydrate metabolism. And this is just an example of one of the branch points. So this particular glucose molecule is attached to 3 other glucoses. And by sprinkling that through, you can make this sort of powder puff of a glycogen molecule. This is a fat cell in which probably 90% of its interior is filled up with triacylglycerols. Proteins, in principle, could be used for a source of energy, but most proteins have a job to do. And so proteins are not typically used as a source of energy except under certain circumstances. So, for example, under stress conditions, the 2 most common stress conditions would be fasting. So if you go for more than a certain amount of time, protein will be drawn down to meet the energy needs, or during fever. And this is one of the reasons why older folks who are extremely lean can sometimes be at risk for a fever, because they don't have anything to energize that fever, and they draw on their proteins. Because we have this slightly compressed time before the next test, what I thought I would do is mark in this chapter some of the structures that I do expect you to know, so you can start having them absorbing into your minds that much sooner. These are all parts of metabolic pathways in the next 2 chapters. One of the interesting things is that we need to be able to make all the components of nucleic acids, nucleotides. And that probably makes sense, that anything that's as essential as your cell, as your very genes, you've got to be able to make, because you've got to be able to replace cells in the body, and we can. And the precursors to those nucleotides come from 2 general directions. There are carbohydrates that are precursors for that, either from the diet or from our own composition. And amino acids. There are several amino acids that we'll see that play structural roles in building up those, the bases. Amongst those, the carbohydrates, glucose 6-phosphate is going to turn out to play a role in a large number of connections. This is the ring form of glucose. We mentioned earlier that there's both a ring and a linear form. This is the ring form. And this is a 6-phosphate because this is the 6 carbon right there. It's the 1, 2, 3, 4, 5, 6 carbon phosphate. This is ribose 5-phosphate, and you probably remember that from our discussion of nucleic acids. Now, there is an interesting twist on getting energy out of these storage forms. I've mentioned hydrolytic reactions for a number of things. When we store the glucose in glycogen, we don't get it back out by hydrolysis. We use something related but different called phosphorylasis. And you can see that the name looks similar, and it means splitting through phosphate. Glycogen is important because the brain likes glucose. And so maintaining a certain level of glucose in the body is key, and controlling glycogen synthesis and breakdown will turn out to be one of the central parts of metabolism too. Here's a picture of what phosphorylasis looks like, and we'll see it in more detail later. Here is part of a glycogen polymer. Here's a free inorganic phosphate. This is inorganic phosphate, and it's going to be added across that bond. Instead of adding water there to split it, we're going to add phosphate there to split it, and it produces a glucose 1-phosphate. So we've created a phosphorylated sugar without any ATP, which is a cute trick, and we'll learn more about that when we get to the carbohydrate chapter. A second source of energy that can be recruited is the fatty acids, and those are through lipolysis. And it's interesting to note that our muscles are actually pretty good at using fatty acids for energy. They're not solely committed to glucose the way the brain prefers. Is this too loud for people? No? Okay. I'll keep moving then. All right. Under sufficient stress, though, under the most extreme conditions, we will break down protein for energy, but pretty much only in the fasting state. Fasting state in this case means 12 hours or so since the last meal. That means in the morning when you wake up, you're getting into the stage where you're your body's getting ready to start breaking down protein for energy. And that's one of the reasons to eat breakfast, to break your fast, spare your muscles, and keep that whole metabolic thing in nice balance. Proteins are broken down for the most part in two ways. There are lysosomes, which are single membrane-bound organelles that act like the stomach of a cell and eat up stuff. And there are proteasomes, which are a protein assemblage shaped sort of like this. And if a protein has ubiquitin added to it, the ubiquitin acts like a kind of a zip code and delivers that protein to the proteasome where it can be degraded and provide amino acids for reuse or for energy. These two things will break proteins down in general, but the rate at which they'll do it will accelerate if you need to use proteins for energy. Now, one of the things that unifies metabolism is that there are certain things that are participants in a large number of pathways. They are common intermediates. They are intermediates, molecules, that are common to more than one pathway. And so they connect these pathways. And they allow these pathways to interact, and the regulation of the pathways will then be appropriate for different metabolic states. This particular set of compounds are ones that we're going to see in a large number of places. Here we're going to feed glucose into the glycolytic pathway. Partway through, we're going to have glyceraldehyde 3-phosphate. So glyceraldehyde, 3 carbons. It's an aldehyde, so it has the carbonyl oxygen there, and 3-phosphate. Many times the names will help you with figuring things out. Not always, but many times. Another common intermediate is pyruvate, again, a 3-carbon compound. It's fully oxidized at one end with a carboxylic acid, and it's fully reduced at the opposite end with a methyl group, which is going to turn out to be important. And this is a precursor for making acetyl-CoA in a step or two. These compounds are not just part of carbohydrate metabolism. We're going to see them again in amino acid metabolism. We're going to see them in photosynthesis as part of the capture of carbon dioxide and the production of glucose. We're going to see them with the citric acid cycle, how the cell can maintain enough intermediates to keep citric acid cycle going. And we're going to see them in fatty acid metabolism. So these are really big-time players in a lot of contexts. Here's an example of some of the things that pyruvate can do. If pyruvate exchanges the oxygen for nitrogen, you can have, let's see now, how can I put this? Let's just take a look here structurally first. You can see here's pyruvate as we just looked at it, and here's alanine. And alanine and pyruvate look rather similar, except that one has a amino group here, and the other has the carbonyl carbon. And there's a simple reaction that will allow us to exchange nitrogens on and off of that carbon. And that's going to be a way to provide for amino acid formation or to refill the citric acid cycle. And we're going to see why that's important in a second. Now, there's another cute trick that pyruvate can be used for. So here it's the 3-carbon backbone. If we invest some energy through ATP, we can actually fix a carbon dioxide molecule onto it and make a 4-carbon oxaloacetate. Oxaloacetate is itself a member of the citric acid cycle and is another one of the structures that it's worth learning. That enzyme is called pyruvate carboxylase. So a fairly logical name. Now this is a mini metabolic map. My husband has always told me I should get one of those huge metabolic maps that has 1,000 compounds on it and put it up and say, as long as you know this, you're in great shape. I think that's cruel. You've noticed I haven't done it. He actually has a PhD in biochemistry. Mine's in cell biology. But this metabolic map is one that's simple enough for everybody to grasp, and you might actually want to redraw it yourself to fix it in your head, because every bit of this is going to give you context for what we're studying later on. And so if you think about taking in glucose and feeding it into a pathway, is there something you'd like to share with us? Okay, thank you. Feeding it into a pathway like this, we've already touched on the structure of glyceraldehyde 3-phosphate and pyruvate and acetyl-CoA. Glyceraldehyde 3-phosphate is another place in which carbon dioxide is going to be related. It itself can be fed off into the production of triacylglycerides. Acetyl-CoA can be used in production of fatty acids, which combine with the 3-carbon backbone. The pyruvate we've mentioned can go to oxaloacetate, which in turn can go either to the citric acid cycle, as I've mentioned, or to amino acids. And eventually that acetyl-CoA will be burnt completely to carbon dioxide in the production of energy as another use of it. So this is sort of a core to the metabolism we're going to be seeing over the next few weeks. Sort of like when you're going to Chicago, you start at the state level before you get down to the street level. One of the things that happens a lot in metabolism is oxidation reduction, loss and gain of electrons. And catabolic reactions tend to be oxidative reactions. And you might want to refresh yourself on table 1.3, which shows you the different oxidation states. One thing to remember is that during oxidation reduction, there's always something that holds on to that electron. There are no free electrons in biological systems. So when there's some metabolic fuel, which is in a more reduced state, if it's oxidized for energy, the electrons that it's going to give off are going to be picked up by a cofactor that then becomes reduced. And that cofactor will be used in some way to generate the energy we want. This is one specific cofactor that turns up in many, many places. This is niacin, one of the reasons you need your B vitamins. And this is the whole structure of NAD, or nicotinamide adenine dinucleotide. Here's the niacin part. This is going to turn out to be what we call the business end, nicotinamide end, I'm sorry. This is what we call the business end, where electrons come and go. But you notice that it's linked through 2 phosphate groups to an adenosine over here. And when we look at these cofactors, it turns out they come in 2 flavors, with or without a phosphate down there. The phosphate down here is about as far from the electron as it can be. It's only acting as a tag, as sort of a post-it note to tell the cell how to use one or the other form of it. And so this is looking at that nicotinamide end in a little more detail. Here we see it in the oxidized state. It can pick up then hydrogen and 2 electrons to become the reduced state. If you look here, this carbon has a bond to a hydrogen, so it's a bit reduced there. Here it has it to 2 hydrogens, so it's more reduced there. This is probably the most common of the electron carriers. NAD plus or NADP plus, either one of those are the oxidized forms of that. NADH or NADPH are both the reduced forms of it, and we'll be seeing that in many times, in many contexts. Another electron carrier that is important is lipid-soluble. NAD, as you can imagine, or NADP, if you look at it, is something that looks like it should be water-soluble. It is. Ubiquinone is lipid-soluble. It's got this prenyl tail to it, and then this interesting quinone ring. And what's kind of handy about ubiquinone is it can carry either 1 electron or 2 electrons. And so it comes in 2 different reduction states that can be exploited along the way by the cell. So when you take in fuel molecules and you use them to reduce NAD plus to NADH, or whatever, what do you do with those electrons? Well, some of it's going to get used for energy. Oxidative phosphorylation in the mitochondria will provide ATP for other things. And some of it will be used anabolically, that since the more complicated things tend to be in a reduced state, if you want to take little simple things and make big complicated ones, you often have to do some reduction along the way. And so those reduced cofactors will contribute electrons selectively and allow you to make more complicated things. In this case, what we're looking at is the electron transfers of oxidative phosphorylation, where the electrons are transferred to oxygen, oxidative, and the end product in that case is water. So whenever you burn sugar as a fuel or fat as a fuel, you will produce water as a consequence of that. So this is the big picture. This is the overview of what we're going to be talking about for the next several chapters. And the general strategy is going to be the same no matter what the starting polymer is. You're going to have large, complicated molecules up here, proteins, nucleic acids, polysaccharides, triacylglycerides, not so complicated. They can be broken down from monomers or made from monomers. Those monomers, as you can see listed here, in turn, can then be broken down to 3-carbon, 2-carbon fragments. That process of breakdown tends to produce reduced NADH. So this NAD comes in and is now a reduced NADH. In the case of amino acids or nucleotides, there may be ammonia that can get reused. Those 2 and 3 carbon fragments are also going to get fed into the citric acid cycle, which is going to produce more NADH for energy. And in the process of that citric acid cycle, carbon dioxide will be given off. In anabolic processes, carbon dioxide can be fixed through photosynthesis into small intermediates, and those small intermediates can be built up into monosaccharides. And we'll see how that gets done too. The NADH that is produced and QH2 that's produced in burning various fuel molecules is going to go into oxidative phosphorylation. Oxygen will get consumed, and ATP will be produced. The NAD plus and Q that's released at the end of that can then be reused up there and go through the whole process again. So this is the big picture. You might, again, want to draw this one out by hand to just kind of fix it in your mind. There is actually in the world a huge amount of metabolic variety. Organisms may have more pathways than we do. They may have fewer. There are pathways where only this genus has that pathway because it's making some crazy toxin or something. We as mammals need as many vitamins as we need because we no longer make those cofactors. But obviously there's organisms out there that do make those cofactors or we would be dead. Plants, since they don't eat other people, have to, in general, make their own cofactors, and that's one of the reasons why plants are a good source of them. Here's a couple of these cofactors we'll see playing roles later on. Ascorbic acid, we are among the very few mammals that can't even make ascorbic acid. I believe the other one is greyhounds. So most organisms have continued to make it. Let's think a little bit about energetics. We're going to see in the course of metabolism compounds or reactions that can or cannot proceed spontaneously. And whether or not they can is going to be a matter of interest at many stages. So you need to think a little bit about energetics. Just to refresh your memories, because I know you've seen this before, if you have some reaction of A and B that go to C and D, if you allow that reaction to go to completion, to go to equilibrium, until nothing more is changing, you can describe that with an equilibrium constant. There are some constant numbers such that the product of the products at equilibrium over the product of the reactants at equilibrium is a constant number. If you have more B, there'll be less A, and so on. But overall, that product is a constant. So we have a number that tells us something about the tendency of a reaction to go to completion. when a reaction is proceeding, it may or may not release energy. It may take up energy. It'd be nice to have a way to talk about that, to measure that. And we can talk about free energy changes in a number of ways. There is a number that has been defined as a standard free energy change, and this is been defined so that we can compare many different reactions. A standard free energy change is the amount of energy released in going from standard conditions to equilibrium. And this is gets printed in this format of delta G, so the change, capital D here, for change of free energy, 0 prime, of standard free energy of biochemical kinds, turns out to be related to the equilibrium constant. So the free energy change from standard conditions is going to be inversely proportional to the temperature and the natural log of the equilibrium constant. Now, it's 0 prime because chemists use standard conditions of, I believe, pH equal to 1. No biological system operates at that. So we use pH 7 as biochemists. Now, the other aspect of standard free energy changes is they start with 1 molar reactants. You never, well, I shouldn't say never, you rarely see 1 molar anything in biochemistry in real cells. And so we need to relate the actual free energy change, the free energy you can expect to see in a context of a living thing, to the standard free energy by taking into account the actual concentrations of the compounds. And we can do that with, we can describe that relationship this way. The actual free energy, the free energy in the context of actual concentrations, will be equal to the standard free energy plus another term that takes into account the amount of the reactants and the amount of the products. Now, one of the handy things about biochemistry is that you can have a reaction that has a large negative delta G, that it's strongly favored to go in the forward direction. And if you can couple that to another reaction that has a positive delta G, it would not tend to go forward. If the sum of the two is negative, the more negative one will draw the other one forward. And one place where we see that frequently is in reactions that involve ATP. That ATP provides an intermediate that allows us to couple unfavorable and favorable reactions. And the structure of ATP is kind of nifty and actually makes it wonderfully adapted to doing that. If you take a look at these phosphoanhydride bonds, so there's a phosphoanhydride bond here and here, as if you'd taken a water out of it. That's why it's an anhydride bond. Look at all that negative charge. Part of what makes them full of available energy is that's pushing apart. And they have a large negative delta G for hydrolysis, which can, if used appropriately, can be coupled to drive other reactions. And this is just one example of it that we're going to talk about in more detail later. You can take glucose. If you want to try to add an inorganic phosphate to it, it would have a delta G of plus 13 or so kilojoules. The hydrolysis of ATP has a negative 30 kilojoules. And so if you can somehow or other couple those two reactions to each other, then going from glucose and ATP to glucose 6-phosphate and ADP, now we'd have a negative 16 or 17 kilojoules. It would become a favorable reaction. So this is one of the jobs of enzymes, is to couple favorable and unfavorable reactions. And this is just a schematic showing you how ATP can serve. If it's produced by catabolism, it can be then used by anabolism and then recycled indefinitely. Someone has said that you make your weight in ATP every day, which is kind of a neat idea. Fortunately, not all at the same time. And this is just a reminder that the concentration of the reactants is going to always be important, that the actual delta G depends on the actual concentrations. And in some cases, you can make it spontaneous by jiggering the concentrations. but there are some reactions where the substrates are not soluble enough that you could ever do that. So ATP, why is it such a neat molecule that it should appear in so many contexts? I've mentioned the charge repulsion, that there's, when it's hydrolyzed, those charges are separated from each other and it's easier for them to sit around. There's also resonance of the products. So here is the terminal phosphoryl group on ATP, and you can draw its resonance forms like this, whereas inorganic phosphate is, can be represented this way. And so you can see that there's a more resonant structure to the free inorganic phosphate than to the phosphoanhydride phosphorus. So both of those things are going to favor the hydrolysis of ATP. Fortunately, that reaction is not a very simple reaction to happen, and so it doesn't happen very fast. It is very favored thermodynamically. The negative 30 kilojoules, it's very favored thermodynamically by its delta G. But kinetically, how quickly it will happen in the absence of an enzyme, it's not particularly favored, and that's why you can have a solution of ATP for minutes and hours and days and weeks. The free energy would be released if it hydrolyzed, but given the particularities of its structure, it won't hydrolyze spontaneously as fast as you might think, given its large delta G. We're going to see some other energy carriers. One common kind are reduced cofactors. I've mentioned NAD already, and ubiquinone. And so one common strategy that we'll see is you have some substrate that's relatively reduced and a cofactor. The electrons from that substrate will be used to reduce the cofactor. And now that that cofactor has been reduced, it can reduce the second substrate. So in effect, the cofactor has allowed the electron to move from this substrate to that substrate. There's no way for there to be free electrons in biological systems, so the cofactor has acted like a bucket brigade to get it there. There are phosphorylated compounds that have high energy bonds, speaking colloquially. Here's phosphocreatine, which you may know about if you read the bodybuilding literature. And then this phosphorus is sufficiently high energy, you can make an ADP into an ATP. All of the nucleotide triphosphates have comparable free energy changes associated with them. So GTP, CTP, UTP, all of those compounds have the same associated free energy, more or less. There's an enzyme called nucleoside diphosphate kinase, so that if you have UDP and CDP, both of them as diphosphates, it can move one of those phosphates over, so you now have a CTP and a UMP. So the first and second phosphates from the end are the high energy phosphates, and this enzyme can move one of those over to generate a triphosphate. Something that we can't do, but microorganisms can, is make a high energy polymer out of phosphate called polyphosphate. And if you think about the structure of the tail of ADP, if you were to make an indefinite number of phosphates, the terminal phosphate would always have a high energy bond by the same reasoning that we think of when we talk about ATP. And when microorganisms find themselves in a location where there's a lot of phosphorus, but not enough nitrogen to grow, they're very likely to pick up the phosphorus and store it. Sort of two birds with one stone. They store some energy and they store some phosphorus. So all of these things play into the actual energetics of what we do on a daily basis or on special occasions. We can see different fuels used in different ways or different pathways used in different ways. Some of these energetic things are freestanding and don't require oxygen. Others are dependent on the presence of oxygen. We call them aerobic. The amount of ATP we have at any one instant is enough to last for about 3 to 4 seconds. So things like the high jump, the shot put, tennis serves are all going to be done from the available concentration of ATP at that instant. We can keep going for a little longer using phosphocreatine cells. That will keep going for, say, 6 to 8 seconds. and phosphocreatine contributes to the energy balance needed for sprints, stuff like that. If you want to go for more than just those few seconds, glycolysis is an anaerobic pathway that will produce a modest amount of ATP. We see that, for instance, you can see here in various kinds of things that last for a minute or so. We can burn the glucose for energy anaerobically for a modest period of time. And then we start to build up the end products and have to do something with them. And we'll see where crocodiles come in a second. Oxidative phosphorylation, on the other hand, can basically continue until you've burnt up all your fuels. And how do these things connect to these very stuff? Well, white meat on a bird, for instance, is something that is used for rapid bursts, but not for sustained flight. That most domestic chickens don't actually go anywhere. Their breast meat is white, but their legs are dark because they have to actually stand up. And so those muscles are doing an ongoing task of standing. If any of you have gone hunting, you know that the breast meat on wild birds is often dark, and that's because they've got to fly somewhere. Ultramarathoners are obviously going to be aerobic exercises. And when we get into some of the other parts of metabolism, we're going to see that at some point you run through your carbohydrate stores and switch over preferentially to your fatty acids. And it's interesting that in the ultramarathoners is the domain in which women are starting to overtake men. We have larger stores of fat. And as a consequence, when you get beyond a certain distance, it actually starts to be an advantage. Crocodiles are interesting in that they are capable of short spurts for intense spurts for a short time. That's how they get their prey, is they'll sit around waiting, and then they'll be real active. And what they'll do is they'll do a burst of glycolysis that's very, very intense. But that burst of glycolysis will be so intense that they will build up end products that they then have to spend half an hour to several hours metabolizing to get rid of. And so it's kind of like a spring-loaded something. They have to, they can do it for brief time, but once they've done it, they're going to have to pay off that oxygen debt, and that goes more slowly. Another compound that we're going to see in a number of contexts, many contexts through metabolism, are thioesters. This is S there for a sulfur, so it's a thioester instead of an ordinary ester, which would have an oxygen there. And the compound that gets often used in these thioesters is coenzyme A. It again has an adenosine in it, which is interesting. Adenosine pops up in a lot of these. But the business end of it, the place where stuff actually happens, is up here on the sulfur. And this interior part here is pantothenic acid, which you may recognize as another B vitamin. So this sulfur here is where the actual thioesters will be formed. This thioester bond has an energy comparable to the energy of an ATP, for reasons that, to my mind, are not completely understood yet. Is that correct? That the high energy bond of a thioester is not completely understood even yet, why that's true. But because it has comparable energy to an ATP, it can be used both for biosynthesis and for energy production. So, despite the travails we've gotten to this point, have a good weekend, and I'll see you all Monday.